Yeah. Way to your non-existent heart, because corals don't have hearts.
That's true.
Yeah, but
Do
hagfish.
sea stars? Sea stars don't either, do they?
No.
No, I don't think so.
No hearts.
Hagfish have all the hearts.
No hearts involved. Hagfish have all the hearts, yeah. There's an interesting sea star on the floor there, too. If we can just take a quick, yeah, just a quick look.
Sure thing. All right, video, go ahead and push on in there, please.
It's got some long arms.
Does. Hey there, buddy.
Yeah. I'm always curious where they're going. Are they looking for something to eat? Are they
Hunting.
It looks like he's
Where was it? It was
hanging out.
Isla de Cocos. They set up a camera on the seafloor and did a time lapse of a bunch of sea stars and realized that they were actually, like, if you speed it up, they appear very much more like predators.
Yeah.
Really?
Yeah.
Ooh.
All right, Alexis, is this good for you?
That's
here?
great. Thank you.
All right, go ahead and come wide there, Marley.
I changed your sea prints to 20 meters.
Thank you. Sometimes I think it'd be nice to have like the tri-tech up there, but
Well, at one point we didn't have that, or most of that.
it's a lot of that to not have.
Yeah, so sonar would be on that screen over there. And then we didn't have the sea prints either.
You might have angled this a little bit more.
Yeah, I don't know why that's angled towards me.
So we kind of came into some excitement and didn't get intros in.
Good call.
Good call.
I thought I heard Holly was trying to, you were going to pitch
Coming up.
a format here.
Well, I just figured it'd be cool to hear like what people's like background and either expertise are, kind of like what's your field?
What's your spiel?
Yeah.
What's our shtick? Okay.
Yeah, what's your shtick?
What do you do when you're not on a ship?
Sure. I'll kick us off. I'm Brandon Rodriguez. I'm a Science Communications Fellow. I work at NASA's Jet Propulsion Laboratory, and a background in chemistry and also in education.
I'm Marley Parker, the video engineer. I'm also a photographer, videographer, science writer, geology enthusiast, and yeah, lots of things, whatever.
All the things.
I'm
All the
self-employed. But that means I get to do cool things like this.
My name is Greg Diffendale, and even when I am here, I work as a day trader.
What are you doing while you're here?
Day trading.
Is that all? Great.
That's it.
Yeah.
All right.
Send your stock tips into. No, don't do that.
Don't do that.
Don't do that. I'm kidding.
Let's make Brandon's life a little bit harder there, that one. I'm Jess Sandoval. On the ship, I work on with ROVs, and when I'm not here, I work on my PhD in material science and focusing on biomaterials. My background's more in biological engineering and then mechanical engineering and now material science. To you, Ben.
I'm Ben Grassian. I'm a PhD student at the University of Rhode Island, and let's see, I use imaging and acoustic sensors and platforms to study the fine-scale dynamics of midwater assemblages, and out here I'm the navigator.
I'm Megan Lubetkin, and I'm on data and I'm one of the science managers out here on Nautilus. My background is in submarine volcanism. I also like many other things.
And recently graduated.
I recently graduated from URI's Graduate School of Oceanography.
And then I'm Holly Richards, and I'm in the science chair, but if you could put in parentheses next to it, science enthusiast chair. So I work with the U.S. Fish and Wildlife Service, and we're the agency that manages, in partnership with NOAA, the Pacific Remote Islands Marine National Monument, which is where we're doing these dives. And so I do science communications for the agency. And I have done science communications for a lot of different land and science agencies. So I'm professionally excited about science.
And I am Alexis Weinig. I'm in the watch lead chair. I am a PhD student at Temple University, and I study deep sea corals. Actually, mostly do work with experiments, so running live coral experiments. But when I come out here, I get to try my best to ID things as we see them, and help everybody guide the dive. Ta-da!
What a team.
I feel like we need a team name.
I was going to say next time we do introductions, we should mention like the most recent but favorite donut that we've eaten.
Okay, I got mine already. I'm ready.
Favorite donut.
I know why you're doing this, Greg.
Also, probably the most fit of all of the teams on shift, I gotta say. The only,
Wow. I don't know, some of us are bringing that average down.
no, the only person I haven't seen actively working out on this shift is Jess, and that's because she was hauling a submersible into the ocean.
Yeah, I don't hit the gym as much on the ship. I usually just sweat it out on deck and then then fall asleep from the heat. Yeah. No, I dance a lot though, so when I'm not here, I dance.
What kind of dance?
Really anything that fits in the mood, but usually salsa or tango.
Cool.
But different space though.
In the science chats, we have like someone is asking about perhaps some drone shots of the island. I don't have access to drone footage out here on the ship, but I can get in touch with some folks back in the office and see if that isn't something we can share. We only recently started, we don't get out here very often, so the Fish and Wildlife Service only gets out to Baker Island or Howland or any of the Pacific remote islands every couple of years to do rapid biological assessments. And we just started adding in doing some drone evaluations with that as well.
Fish.
So we may have some drone footage that we can share, or if not, I know we have some great aerial shots and landscape shots of the islands to give people a sense of what we're looking at.
Marley, you want to start fishing in there and we'll frame up on the fish?
Is that the eel that's not really an eel?
I don't know. Fish are not my specialty, and they are, I feel like, very tricky to ID. I often think it's one thing, and I find out later that I was completely wrong.
Yeah, Greg, I think you're talking about a cuskeel.
Yeah.
Yeah, it looks like maybe a cuskeel. Just a type of fish. Deceptive name.
Enjoy it when the shrimp just sort of fall through the screen.
Jess, I'm going to zoom just a little further.
Sure.
Yeah,
thing,
some of the
go ahead.
previous work. So the only other ROV dives on Howland and Baker Islands are by the Okeanos Explorer, the NOAA ship that was out here in 2017. And they did one deep and one shallow dive on each island because they operate only during daylight hours, so their ROV dives are much shorter than what we're able to do with the Nautilus.
Marley, you want to go ahead and come white, please?
But they did see pretty diverse fish community. So they had some people on their team that were able to ID fish pretty well. And I think there was, on one of the dives, 38 different species of fish.
Ben, how far have we gone so far on this dive, and how much farther do we have to go?
You guys see the little purple blob down there? too?
Yeah, it looks like a holothorian.
Sorry. So, since our watch change or in total?
No, just in
Yeah,
total.
let's see.
How far do we have to go on this whole dive there, Alexis?
The dive total,
Yeah, total.
we're trying to get to 400 meters.
So just as a vector length, it's about 2.5 kilometers. We've come,
Probably like 100 meters.
oh actually no, sorry. We have two maps we're using. One that we ourselves just mapped with our shipboard multibeam and a previous mapping data. So, yeah, so in total this dive is going to be about a 5 kilometer transect.
Come up a bit, Greg.
We've come about 1.8 kilometers.
Holly, you mentioned communicating with some scientists back on shore. Can you talk a little bit about who's listening in and how we're trying to support that community?
Let me just ask Ben a question real
Sure.
quick. Ben,
Sure.
did you say we've traveled 1.8 out of the 5 total?
What did I say? Let me just measure one more time.
Sorry.
No. So I'm giving you just like a line segment.
Yeah.
But we've kind of meandered around as we climb up this ridge. But we've gone about 1.3 kilometers so far.
Okay.
Do we want to go 5?
In total for this dive, our last waypoint is about 5 kilometers from the launch.
So we might want to pick up the speed.
Yeah, if we want to,
Yeah.
especially as we're,
So are we planning to come off bottom at
3?
3 a.m.
Okay. All right.
All right.
Yeah, I'll increase our steps.
Yeah, I can do 40 meters then.
Great.
Yeah, we have a pretty extensive dive track for this dive. Again, the previous ROV dives out here were limited to a much shorter time, so we wanted to spend a little extra time covering a bit more ground. But it can be difficult sometimes to just keep up the pace, especially if you're stopping for sampling or you're changing direction based on where it looks like the hard bottom is.
Did you want us to, I know at least last leg we were kind of keeping one heading, and then that helps you guys with the visual transects when you guys are processing the videos. Would you want me to just maintain a single heading?
No, it's not. That's not necessary.
Okay.
Yeah.
He just probably wasn't talking to SPL.
Can you hear me now? I was just talking to the bridge. Okay. Testing.
Oh,
testing.
there's another Dumbo to the right.
Oh yeah. Good eye.
Oh, hi.
You guys want to look at it while we wait for the ship?
Yes.
They're located in such indiscriminate places, I swear.
Yeah. It's like they have camouflage.
Oh, there's a fish, too.
Yeah.
Which would you prefer, the
And
fish or
it's-
the Dumbo?
Something white up on the rock in between the fish and the
It looks
Dumbo.
like
It's a coral stem.
A coral base, yeah.
Yeah.
We can
Let's look at the
fish.
Dumbo. Sorry.
Oh, no,
Sorry,
go ahead.
just
You make the call.
let's look at the Dumbo. The fish is a halosaur, but the Dumbo has a weird.
Bridge or
I'll
something.
turn off the laser so I
Line
don't
on
There's
it.
a weird
shoot his
shape
eyes.
there.
All right, go ahead and start pushing in their video.
Looks like it
Wow.
has,
Huh.
Been injured?
yeah, but healed.
Oh.
Poor baby.
I wonder what it would
Yeah.
have.
What?
Pretty sure it's
Who
sleeping.
did that?
Yeah.
Yeah, I turned off the laser so we don't
Yeah, that's good.
get them more mad.
Oh,
It looks like Venus or something like.
That's
How
gnarly.
interesting. Yeah.
yeah.
I mean, I think I've read that their predators are sharks,
Yeah.
but wow.
That's
I mean,
big
that's
scar.
a pretty hardcore octopus. If it got
Yeah,
that.
right.
That's
I
an
was gonna
intense
say,
scar.
like, if it's
Now the way he's looking at me is a little threatening.
Yeah.
Dang.
He's seen worse than us.
I would not cross that octopus.
It's really neat, actually.
it's so interesting that how different the color is. Like,
Mm.
what would have just
From
like
scar tissue that
Right.
would be on us, you know.
Yeah.
But it's a darker version of his pigment. Yeah.
All right, Marley, you want to go ahead and come right there?
I mean, it's kind of just a good reminder of how tough you have to be to live down here, right?
Yeah.
This is a very extreme environment.
True, but it's not often represented as a scar across your face, right?
Yeah.
It's, this guy's got a story.
Come on up.
Makes two of us.
Raj.
Every octopus has a story. Something.
where?
Thinking. Sinuous down there.
A little fish.
Yeah, a little fishy.
There's a coral up here. Would you like to go look at that?
Yeah, just quick
Quick zoom.
zoom.
All right there, Marley, go ahead.
Looks like another primnoid octocoral, so has these polyps that are facing
Downward.
downward, right?
Yep.
It's not very planar, so it has a lot of different directions that the branches are going.
Is that good for you, Alexis?
That's good, thank you.
All right, go ahead and come right there, please, Marley.
It's interesting to me all the diversity of the coral polyp morphology when it's not like they're inducing flow around themselves. Like, what is that diversification in terms of like foraging success?
That's a good
Ooh,
question.
Yeah.
That's a really interesting question.
Because there's this concept, at least in the plankton, where it's like, how do you have so much niche separation when you're in this kind of relatively homogeneous environment fighting for like similar resources? And to me, I see the deep sea in terms of the benthic
A
environment
lot of
similar.
corner.
What is that?
What do you see? Oh, sponge.
Oddly shaped something. Probably a sponge.
Or a
Conopoduncle.
holothorian, maybe?
Holothorian?
Sea cucumber?
It's like a holothurian, I think.
Yep.
I'll do quick zoom. You want to go ahead and push it in there, Marley? Marley?
Pretty cute.
Sure is.
All right, one final push and then we'll
Just
let it
crawling
go.
along. So sea cucumbers are
That's
a
great right there.
echinoderm, so they're related to like
I'm
sea
not looking
stars,
straight down
brittle
yet.
stars,
Roger.
but they don't have
Yeah, I need to start going pretty soon now. All right, Marley. We're
they're
just
just
doing a quick zoom
processing
on this guy.
sediment
Go ahead and come by,
essentially.
please.
So they suck sediment in their mouth,
Now I'm straight down.
take all the nutrients out, and out it goes the other end.
Yeah?
Yeah.
There's like there's some species of land crabs that'll do that too. And so they'll just like eat all the sand on the beach, clean all the diatoms off of
Yeah.
it, and then poop out these little sand pellets that are
Yeah.
cleaned of all nutrients.
If you ever see like coils of sediment, that's a
Start
Sea
pushing up the
cucumber
hill.
poop?
Looks
Yep.
like there's a
Yeah.
big shadow there to your upper right.
There's still about 20 meters left in the ship move if you're...
Yep.
Okay.
I would just climb and push out faster.
Yeah. That's what I'm feeling. I'm feeling that.
Because remember, the ship's also moving faster now than it was before.
Sorry?
The ship's moving faster than it was before because we're doing larger steps.
Yeah. Oh, yeah, definitely.
We're just getting closer and closer to the island, right?
I think so. Yep.
Mm-hmm.
Are we still good with the 40-meter moves, or I can...
You want to do 30?
30, okay, great. That's...
You want to stand by for a sec?
Yep.
I'm going to push out in front.
Yeah, and your gain's at like 60.
Yeah.
uh-oh.
Oh, no. Dang.
Somebody coming in there.
I have a question for maybe Holly and Megan. You guys probably both have info about this. So like we're looking at volcanic rock underwater and we're trying to understand what inhabits that space this deep. But we have volcanic rock and soil in terrestrial land as well. What kind of, do we see communities that aggregate on those rocks?
Do you want to?
Why don't you take the sub area? I can comment.
Sub-aerial. As opposed to terrestrial. Yeah, I mean, it really depends on the environment where that volcano is taking place. So if you look at like the Hawaiian Islands, which most people associate with as being like super lush, one of the other key factors of the Hawaiian Islands is that it's in a tropical environment with lots and lots of water, so lots of things to break that rock down into soil. And if you go onto, say, the Big Island, where so the most recent volcanoes are, including very, very recent volcanoes, the first things to colonize or to start growing in that volcanic environment would be the ohia tree. And so you have these sort of, you know, primary species like ohia, or if you're in the southwest, it's usually sometimes juniper or aspen will take root in some like lava flows in like northern Arizona. So you have these sort of initial species that sort of make their way and are hardy enough to take root in that. And then as they start growing, the roots will break down the soil, break down the rock into soil, and then it really depends on the environment around it. So in northern Arizona, you'll have 1,000-year-old volcanoes where the lava is still essentially barren because the conditions, like the environmental conditions, aren't right to break it down into soil. Whereas you go down to the Hawaiian Islands and you have fairly recent volcanoes that are already getting broken down into like very lush environments.
Cool.
Yeah.
Yeah, just thinking about it from a perspective of like how this volcanic rock ends up promoting life, you know, and like all of those types of, it doesn't matter if you're underwater or above water, it's really significant for things to start to grow there.
Well, all of, like, we mentioned Hawaii, but, like, all of the Pacific remote islands, marine national monuments, like, these are all ancient volcanoes that are now either islands or atolls that are homes to these massive seabird colonies that now really have nowhere else to go because these you know, these seabirds and these migratory shorebirds evolved in spaces and in environments where they had no terrestrial or mammalian predators. So like albatross and petrels actually, and like a lot of the burrowing birds, or the lesser frigate birds actually that make their homes on Baker and Howland, they'll come back to these islands and they'll lay their eggs right on the ground. And so if they're in a place where there are mammals, like the mammals are going to eat those birds. So they have to have these sort of safe locations. And for them, it's out on these remote islands where there have never been any mammalian predators.
Yeah.
So these are really some of the last safe habitats for a lot of these birds.
Hey, Gabby, can we take a look at that coral to the left? Thanks.
Ooh, Tina 4.
Video, can I get a zoom?
Sure can.
Oh yeah, on the Argus camera.
Yeah.
Would this be a place we could sample, possibly?
Yes.
Cool.
As long as we stop the ship and know soon.
Yes, I would like to sample it if it's possible. We've seen a couple of this type of primnoid. I think it's a Norella, which is the genus of octocoral. But again, having that ID for confirmation, both morphologically and using the DNA is really helpful.
Okay. Can you go wide video?
Coming out.
So you're really excited talking about this island.
I loved it. And like the shallow water corals around all of these islands and atolls, they wouldn't be able to be there if it weren't for the volcanoes that had one, that created that space too.
The volcanoes that create the space, and then actually, so the colonies themselves. So in terms of nutrients that are necessary for those nearshore marine environments, like these seabird colonies are foraging over tens of thousands of acres.
Yeah.
And bringing that nutrients in the form of their prey back to the islands where they're either pooping it out or dying, and bringing those nutrients and sort of collecting them on these islands to then wash into the nearshore and the coral reef environment, so in terms of nitrogen.
Yeah.
Yes. Yeah. You do one.
That's cool. We just come for relief and we get the sample.
Yeah. Thanks, guys.
You're welcome.
Well, to be fair, we left them with a sample.
Yeah,
True,
it's true.
it's
One
true.
for one.
Yeah, that's really why I requested it right now.
So, do we have a number for this sample?
Yes, this is 25.
I don't think bubbles, yeah, it's not long enough.
Can we just get a zoom on it before we sample it?
I'm all the way to the right with the camera.
So go for zoom.
Okay.
That's as good as you're going to get there, I think.
Cool.
It's shaky.
Yeah, we're in real tight.
Something funny going on there.
Oh, you think?
Maybe just the way the auto heading is trying to go somewhere, maybe up against the rock.
Uh-huh.
Whatever. Doesn't really matter.
Could we just pan down a little bit? Or are we?
Do you want to see the base?
Yeah, there we go.
Okay, there
Thank
you go. I wish I could take credit for that. Those are just luck. That worked out lovely.
Okay, that's great. Thank you.
Okay, go wide.
Pulling out.
Yeah, you can go all the way wide, just for starters.
You got it.
So just like 10 to 15 centimeters.
Roger.
So what are you looking for, like, with this sample?
What do you mean?
Like, why did you choose this one?
Well, we've seen
Yes.
a similar
Porch light is off right now.
a similar coral a couple
There you go.
times today. And so just to confirm the ID of it,
Yeah.
we'll take a sample. And we don't have any, the previous dives at this, around this depth, only sampled a couple sponges. They didn't actually sample any corals. So
Can't see.
yeah, these act again as like
That's how you
essentially
know it's the right
vouchers
spot.
to
Yeah.
confirm what we're
Right.
seeing. And I'll talk about it, yeah, a little after.
Where are we going with this sample?
You can put it in the forward starboard side, the B box, or if you want to get it on the starboard bio box, the small one that's forward A would
Okay.
be work.
Try to go forward A, I guess.
No, so starboard A and forward B.
Starboard A, I meant.
Yeah.
Sorry.
Yeah, starboard A would be great if you can do that.
Yeah. Got it?
Ready
Yeah.
for?
Yeah.
Okay. And box out. Oh, and I can switch some cameras here for you.
I think. Yeah.
Still in there somehow.
Yeah.
There
There
it
you
is.
Now it's
go.
out.
Nice. Awesome. Well done.
Nice job.
Oh. Somebody watch your mic.
What?
Somebody's rubbing their mic on something.
Somebody's rubbing their microphone.
Stop rubbing your microphone. Unless it's me. It could very well be me.
Yeah, excuse me.
Calling
it was me.
yourself out.
I'm gonna give you a reset, okay?
Okay, that'd be great.
Yeah, so one of my lab mates, Steve Oskovich, is at Temple University, and he was on a previous cruise out here this summer when they were at Palmyra and Kingman Reef. And a large part of his PhD is about coral distribution in the Pacific. So
Can I bug you guys just
Oh,
for a
sorry,
second? Are we
yeah.
all set here?
yeah, we're good.
Okay,
Thank you.
great.
Okay.
Onward.
40 degrees reading.
Give me just a second to catch up.
Yeah, sure.
And then I will be right with you.
Look how bubbly that lava, that basalt is. And yeah, so he's been using data from 2017 when they were out here with the Okeanos, when we came out with the Falkor in the Phoenix Islands protected area, and now he'll use all of the samples from these cruises as well in the Pacific to really start teasing apart some of the distribution patterns. So a lot of, especially octocorals, can be distributed by depth. So you have certain species that will be in a certain depth range, but they won't be above or below that.
Give me just one more second.
And
this
then
is quite nice looking.
Yeah, that's a nice sponge.
Can you guys give me a zoom?
Push it in.
That is a nice looking sponge.
Ooh.
Glass
I love those
sponge.
so much.
Yeah.
They are fantastic. They're so delicate.
They are. So all those little like what look like hairs coming off are spicules. So they're like little glass shards essentially that make up the structure of the sponge. And they're beautiful, but oh, did you see the shrimp?
Yes.
Yeah.
Yes.
The captive shrimp.
They
hurt really much when you touch them.
right. I was like, we tried not to sample them because
Okay.
They hurt?
Go wide.
oh yeah.
Oh yeah, they
It's
go
like-
through your gloves.
Have you
Oh,
ever
really?
Have you
Oh,
ever held
yes.
like a prickly pear from a cactus?
Yeah.
You know how like the little tiny spines get in your fingers?
I try not to touch this thing.
Well,
Exactly.
it's very
It's
similar.
like a stinging nettle or something.
Yeah, it's like a similar
Or
sensation.
fiberglass.
Yeah, it's like
Fiberglass.
fiberglass. You can't really pull them out.
No.
They're
Yeah.
kind of just in there.
They're in there until they disintegrate, and
Yeah.
it's like itchy and unpleasant.
That
is
sounds
the
like
material?
fiberglass.
It's
glass.
silica.
It is silica.
Yeah,
glass.
it's
Oh,
silica.
wow.
So they're like,
Yeah.
they're made out of silica.
Yeah, often that
Okay.
becomes a
Yeah,
problem
go
when
for
we
it.
collect a rock that has
Reach
like
now, step
little
please.
bit-
little growth on it.
Yeah.
So not intentionally getting a large sample. But
Isn't that all those the DFO scientists were getting? Those was the glass sponges with like all of the spikies on them?
Who wanted it?
The what?
So we did this cruise last year with all these gorgeous giant like trumpet shaped glass sponges everywhere. And we collected a lot of samples. And I don't know how they
Oh, DFO.
yeah.
Yeah, they must have.
They must have like real serious gloves.
Yeah, some,
Yeah.
Just maybe not nitrile, I guess.
Gardening gloves, maybe. would
Yeah.
work.
I've taken to wearing gardening gloves in the shop like all the time. And we have pink ones. Me and Mike
Oh,
both.
nice.
Yeah.
Nice.
They're pretty
Stylish.
we're a
rad.
team.
We
Glove
sure,
buddies.
We're a
we
unit.
sure are a unit.
Sharing a brain.
Gabby is bringing life to the van
Yeah.
with her laughter.
Yeah. Did you mute your laugh, Gabby?
Gabby, someone,
Yeah, she did.
Someone
She's like, I
wrote
can't
in.
take it. I gotta mute myself.
Someone
I
wrote in
used-
and said, even when Gabby tries to mute her laugh, we can still hear it.
Hello, there.
Yes.
They said
really
it in a
thought
very
I
good
was-
way.
I really thought I was getting away with it.
No.
It just becomes like an echo, a Gabby laugh echo.
Very much thought I was getting away with it.
No.
Yeah.
You're way out front there now.
Yep. Sure am.
There you go.
You're
Please.
way behind.
Always.
Do you ever think of that?
Gabby's always way out front. Leading the way.
Pioneering.
Yeah, sure. I'm towing the whole ship with me. That's what's happening
Yeah.
here.
Come on, little Nautilus. Let's
This
go.
ship's actually facing the opposite direction.
It started. It needs its 2 to 3 minutes to start the move. You know that.
Yeah, I know, but I got,
I
you
know.
know, antsy.
Just ready to go.
I have things I want to see.
Yep. Now it's moving. Now
Is that just marine snow?
in
we're
2
picking
minutes
up?
it should be okay.
Some
Yeah,
of it's
just
just
a-
coming off the vehicle.
Right.
When I go
Yeah.
backwards, like anything that we've gotten on the vehicle from setting down will come off.
Okay.
Yeah. There's other places we've worked where it's like really silty, and like anytime you set down, it's just like a sediment plume, like all around you.
I bet that when you move forward, the same happens, but we don't see it.
Yeah. Oh, exactly. And then it matters not.
little fishy.
Albert, can you tell us about what it means when you say you're making a ship move? What does that mean for people at home?
That means that the ship, like this ship, has a positioning system based with two thrusters.
Video, can you give me a zoom?
That lets it move in any direction, and when we ask for a ship move, it
Stop.
means that we ask for the bridge,
kinda
the people
looking
who
at
actually
this.
drive the ship,
Okay, a
to
little
move
further.
the number of meters that we tell them at the bearing angle that we tell them. So we position the ship in a different place, and by that we move Argos 1,500 meters below
Oh,
us.
like dead sponge.
Yeah, looks
Yeah,
like
dead
it.
sponge.
Looks like there's a tiny little
like
sea star in there. Is that what that is?
Oh yeah. Little
Okay, go wide.
penguin. There's also
Take
like a
your
little
bubble camera.
mollusk,
Yeah,
a
do it.
little snail shell up there. We can keep.
It is kind of mind-blowing because we're kind of going through here and essentially moving through this massive space of darkness with a flashlight.
Yeah.
Video, can I get a zoom?
Despite all the data that we're actually getting, which is a massive amount of data, we're still just getting this tiny fragment of the ocean environment around us.
Exactly. Looks like an
More
old,
of the same.
Yeah, old
Base of something.
base of a sponge. And then those little ones look like little coral bases too. And then there's a shrimp.
Very bouncy.
Oh, there's a little tube worm.
Oh, I'm stretched way out. That's why. Go wide. Very stretched out still.
Yeah, and so we're diving now on kind of like the south, where are we? Southeast side
Another step.
of
Yeah.
the
keep it
island.
moving.
Bridge nav step, please.
And we haven't
Thank
seen
you.
a whole lot of coral or sponges throughout this dive so far, but some of that might be, we don't fully understand the currents at these depths. We
Yeah.
don't
Go for zoom.
understand all of the oceanographic influence, and some of
Good.
what might be the case is that, you know, on another portion of the seamount, there might actually be more dense communities, but we just have the opportunity to do one dive here.
Right.
So we're only really seeing this side. I know on the previous dives, they were on another section of the seamount, and it was similar. They had quite a bit of sediment in the area, which also makes it more difficult for some of the sessile or organisms that are like stuck on the rocks to actually live, because they need a hard substrate to settle on.
Okay, go wide.
We'll actually be crossing the equator tonight, I believe.
Go wide,
Or we
Marley.
crossed it?
No, we are
We're cross-
north already.
Oh, we're
Yeah.
north already? We
Yeah.
crossed it
Yeah.
yesterday?
We're
Yeah.
crossing the date line.
Oh, we're
But
crossing
you
it.
can
today.
say, we can say that we're crossing it again today because it's like we're repeating the day tomorrow. So.
I know, that's what I was going to say.
Yeah.
But are we crossing the date line tonight?
Yeah,
then?
so
Okay.
tonight we're switching back to today again.
Yeah, so on the ship for us, it's been September
First.
September
First.
1st and tomorrow September 1st.
and tomorrow is also September 1st.
Indeed.
Yeah, we go from +13 to -11. It's gonna be actually the same
It's
time.
going to feel, yeah, it's the same time.
Yeah.
Same time of day, just
Just a different day.
different day.
Yeah.
We're back in American Samoa time.
Yeah.
Well, we never were in American Samoa time.
Yeah, we were.
When?
A while
Not
ago
in this
we were.
cruise.
Yeah, okay, sorry. I was last
I
week.
was too. So yeah, so there, Albert, we were before.
I
you
was
got
there.
here.
This ship was here.
Yeah.
You weren't on
Before I
it.
was here, I don't know what happened. Sorry, guys. Sorry. I.
The ship
Breach
ceased,
nav, can you
ceased to
step again,
exist.
please? Let me
Yes.
focus in something else.
Just decides to talk to somebody else.
Gabby's going again.
Yeah, who can hear her?
Is it ice cream day today?
Oh, it is very
There's a
nice.
fish-
And tomorrow.
No, no,
Better
no.
Days.
Marlene
What?
said 2 Sundays, only 1 ice cream.
What?
Huh?
That's
not
what
how
she
it
said.
works.
That's not how it works. It's not fair.
You guys need to take it up with Marlene then.
She did also make us donuts today.
And
so I
she
feel
did
like-
make
Oh,
us
those were
donuts.
good.
To make up for the only one ice cream.
The
day.
Wait, but
Donuts-
so are we getting ice cream tonight or tomorrow?
I
A mystery.
Both days.
Listen.
We have to. It have to be both days.
I don't think it has to be. I think it has to be whatever they say it is.
Yeah,
You
true.
could eat enough ice cream for both days.
That's an interesting strategy.
Yeah.
Yeah.
Just take extra. Keep it in your
Just
room.
take it home. Put it in a mug.
It
Take
works
it to your room.
like a charm for ice cream.
Under your mattress.
Yep.
Where nobody else can find it and steal
Well,
it.
Can we
Ben
take a look
sleep
at what
under
those
me,
little white
so
things
he
are?
would find it
Yes.
eventually.
Which little white things?
Just all the speckles on that rock right where
Barnacle
the lasers are.
things.
Yeah.
I just wanna
Okay.
see-
Go for zoom.
If they're old bases or what. Oh, and there's an urchin up there. Oh, there's a little squat lobster in there.
Oh, they look like they're spiky, right? If you look at the ones on the sides.
Yeah. So I think they're bases from corals. So at some point they were growing there and snapped off.
So we've seen quite a lot of them.
Yeah.
Right?
Could it be that they are growing, not they were growing and snapped off?
No, because normally these corals start as like one polyp, so there would be polyps there if they were alive.
Ah, okay.
But it's interesting, yeah. I don't know if it's like there was really high current at some point that just snapped
Can
them.
you go a little closer? Yep. Video. Suppose I could always go a
Breach
little
nav,
closer
step,
too.
please. Thank you.
Have you seen what you want, Science?
Yes, thank you.
Okay, go wide. Must
Oh,
catch
there's
up.
somebody swimming over there. Oh yeah,
Ooh,
do what you
is
need
it
to
Albert? I don't know.
I wish I was swimming over there. Well, not over there, like 200 meters above that.
do. Yeah. We don't want you to die from pressure.
Yeah, no, no.
1388 meters above that. I thought you just said like 200 meters above that.
Few hundred meters.
Albert's fine swimming at 1100 meters.
Right.
Yeah, 1100 is fine. But
Yeah.
1200,
That's getting crazy.
slightly too much.
That's just too much.
What are you, insane?
There's
a
a little
coral.
coral. Or a crinoid, maybe?
I don't know. Maybe.
It's okay if we can't.
I gotta catch up.
Yeah, do that.
I know.
All of a sudden, she's like, I don't have more leash, and then she's behind.
I know.
Come on, Gabby.
Very easily distracted.
Okay, I'm going for dinner.
Oh, fish.
Ooh.
Sorry, guys.
That's beautiful.
It was really pretty. We gotta go.
Yeah.
It's not a vampire squid. I really like vampire squid.
Well, that was a really pretty vampire squid earlier.
Yeah.
Lots
Some
of these
coral.
primnoids, yeah.
Is that pretty much all we've seen
Yeah.
this dive, right? We've seen mostly
Yeah.
primnoids.
There I am.
On our shift.
On our
There
shift.
we
on our
are.
shift. Mhm.
Yes, very not sensible right now. Driving around. A box. So the only one left is D box. Right? The end little one?
No, we got more than that. We got B, E, and D.
Oh, okay.
We're loaded.
D box.
It's another coral.
Yeah. We haven't really seen any cup corals on the last two dives, which is interesting too. We normally see these little, like solitary, so one polyp, smaller scleractinian corals. And I think maybe we saw like one yesterday. We see some maybe as we get more shallow, who knows? Oh, there's a little swimming holothorian. It's gonna be out of our view. It's okay, but
Yeah, we've seen a number of
sorry.
those.
Telling you, that's what we say in the ROV shop all the time.
All right, coming up.
Roger.
And tilting camera up, and we're gonna get back into normal.
but I think it's dead again. I'm kind of surprised, by the dead sponges. I don't really know what would, unless there like used to be a lot more particulate in the water, and now there's not enough to
Look at that
sustain
one.
them.
Whoa, look at that one.
I don't think that one's dead.
Or there's just a big guy in the neighborhood.
Are we
Just like
interested in a
taking it all.
sample?
It heard you talking.
We are
Zoom
going
on in.
to take a sample if we can.
Sure.
Oh.
There's also one
wow.
above it. You can see it in
Oh.
Argus view too. The one above it looks a little larger.
Really?
Yeah. Well, so look at this one. I'll look at the one above.
Cool.
All right,
Look at it,
Ben,
it's so
how much more
beautiful.
do we have in this move?
This is gorgeous.
We have about
So that
20.
is 10
All
centimeters,
right,
those
so let's
lasers.
go ahead and hold.
Which means we're looking at, what, a meter across, probably? But even, like, so that
I'll bring us
does,
back.
Yeah,
see the
go ahead and bring us
sand
back
in
up.
all the
Meters.
folds?
Yeah.
Yeah.
That would not be great for long term for
Right.
that sponge. So
Right.
that does make me wonder if over time they are just sedimented out.
Yeah.
So, like, there's too much sediment and it just
Yeah.
kills them.
Because
not
they're
able
filter
to
feeding,
right.
so having all of the sediment on is going to block their ability
Right.
to like get
Look at and all the
nutrients.
shrimp just hanging out in there.
There's a lot of shrimp hanging out in there.
Science, can you see the larger sponge now in Argus?
Yes.
Just outside Herc's light pool.
Wow.
Hey, you want to come wide there?
Wow.
You want to come wide there, video?
That's really cool.
Marley, you want to come
It
wide,
may
please?
not be as alive.
Yeah, it looks like it's a bit brown on top.
Let's just zoom in. Oh, yeah.
Yeah.
Oh, yeah.
Wow.
So, yeah, it looks a little bit more dead, huh?
But we could still sample it. We could sample the one that's dying. That's fine.
It does, and it looks like there's a lot of sediment in it.
It
too.
does.
Yeah.
Oh, man.
I was thinking when I came up here, I was wondering if that could be one of the reasons why the smaller ones aren't doing so
That's
good. It
what
might
it
be
is.
too much sedimentation.
Right. Yeah, we were just talking about that before we saw the really nice looking one.
Because there's a lot of sedimentation here.
There
a lot
is.
in the water.
But like the edges of that, we could sample. Like on the left, there's kind of some that's white.
Yeah, I think most of it is actually still alive, just covered.
Yeah, yeah, it kind of looks like
Which
that.
means like over time it would slowly suffocate, essentially.
Marley, you're full wide right now, right? Alright, I'm going to look down and I'll sit down here if we're going to sample this guy here.
What's happening, George? What's happening?
What's up, George? You can go ahead and sit there if you want there, George. Better to sit than to stand.
So I'm curious. I mean, they're right next to each other. Why is there so much sedimentation on this one instead of on its neighbor?
The neighbor was throwing the sediment up.
Yeah. It could be the direction that it's slanted. It's a little bit different orientation. Also, it's a bit older probably, so it's had more time. And some of it, the tissue just looks brown too. So it looks like it's kind of just dead in those parts. Oh, there's some like sea urchins in there.
Alright, go ahead and hit that stick block and see how we settle. Just want to make sure that I'm not going to hit the other one.
Yeah, we're far enough away from the other one. You can always increase your Z bias a little bit too. Like leave the stick lock and just kind of crank
Change
it down.
wing. Yeah, that's a good point.
Oh, we broke a rock.
Wow.
Might actually work in your favor.
Come on. Alright, you gonna try that stick lock one more time? Has some lateral going now. Maybe that'll help us out a bit.
Have you sampled the sponge before?
Yeah.
Okay.
Floaty bits for days.
Yep.
Alright, you guys want to take some images of this before we take some samples?
That would be great.
Alright, video, you can go ahead and do your thing. That is some dying sponge.
Alexis, do you want something specific as far as imagery?
Alexis went to dinner, so this is Sam. Speaking for Alexis, no, I think getting some close-ups is like this is great, and then I think we've already imaged the broader picture pretty well.
How much live sponge do we need?
Maybe like a baseball size clump, but also whatever we can get.
Okay. And where are we expecting to put this?
You could shoot for the most aft small starboard bio box.